So I'm very pleased to introduce Professor Tim Maughan from Oxford, who has been working in this area and developing new designs, and particularly in colorectal cancer. And Tim is going to tell us about his work, particularly with Focus 4, which is ongoing at the moment. Tim.
This is going to be a bit of a change in gear, because we've been quite general so far, and we're now going to dive into the specifics. But I hope I'll be able to keep it light enough for you to continue keeping engaged. So it's a great meeting. Thank you very much for inviting me. And it's a challenge to know how to talk about stratified medicine. We're just starting a precision cancer medicine institute in Oxford, or we're building towards one, so I'm pleased about that concept. So colorectal cancer, and indeed cancer in general. So what's been coming forward in the last few years in cancer has been that the realization, as if we didn't know it already, that every patient who walks in to see me in the clinic has a different disease. And not only is that patient's disease different from any other cancer patients I've seen, but it's changing over time. So cancer is unique and it's evolving. So how do we make progress in the clinic with that realization? So we use this term tumor heterogeneity. They're all different. And we need to get from that to be able to find the best treatment for this patient through biomarker selection. The second challenge of that is that as the cancer evolves, clones become resistant to the treatment, and how can we overcome that huge problem, and one that I'm not sure that we will ever overcome. But we're trying to do that with combination therapies and with earlier intervention, and I'll give some illustrations of that. And how are we going to move the field forward in terms of using or discovering and validating biomarkers that will actually help us to find the right treatment for the right patient. And that requires, I think, a period of very broad and intensive characterization using access to the omic technologies that we now have. And in that context, they need to be hypothesis-driven rather than just fishing exercises. So some of this I'll talk about. So a dive into colorectal cancer biology briefly, 3 slides only, won't be detailed. But this is one of the headlines from the Cancer Genome Atlas report of intensive characterization of 224 patients. And what I want you to recognize from this slide is that there's a clear cut between this group of about 15% of patients and this broader group that look very similar. So this was a clear view. They called it the hypermutated group, and you can see that it's associated with something called MSI, status microsatellite instability. And these are associated with abnormalities mostly in these genes here, which are genes of the mismatch repair process. So this is a group of patients where there is a particular carcinogenic pathway. Now that's something we've known about for a long time. It results in these high levels of mutation and is different from the other. I'll leave it there for the moment. There's much more to be said. But as they've dug in to what's different between each cancer, you can see along the bottom here, every patient is this just in one column. And you can see that although there are some patterns, largely they're random mixtures. And we're looking at 5 major pathways that are abnormal in these sorts of colorectal cancer. And as I said, every patient will have a different mixture of those pathways failing in their cancer or being activated, leading through to this highly chaotic system. And yet which generates a disease which, as I see it in the clinic, looks identical pretty much between one patient and the next, although hugely different in terms of therapeutic response. Now, for most of these pathways, we do not have treatments in the clinic. There is nothing against Wnt signaling that's actively licensed in the clinic, some in development. TGF-beta signaling, P53 signaling. We've got something operating down this pathway, and we've got drugs in development in PI3K. So we have very limited utility to be able to intervene in these pathways in the clinic at the moment. So when you look at the first emerging data about from the genetic point of view of colorectal cancer, you see huge heterogeneity. And an effort to understand and integrate this has been going on over the last 2 or 3 years, looking at gene expression. And last summer at the big American cancer meeting, this group, the Colorectal Cancer Subtyping Consortium, gave their first presentation of trying to integrate this on the basis of gene expression profiles and subdividing colorectal cancer into just 4 groups. Is this reasonable? And that first group, CMS1, is essentially that group that was on the left-hand side, that 15% that are characterized by microsatellite instability. We know about that group. This second group up here was what anybody in the field will know as the standard Vogelstein model. That is the way that everybody has thought that colorectal cancer has been developed. It actually only accounts for about 35% of people on this analysis. And what was really interesting about this analysis was these 2 new groups, one of which, CMS3, which is an epithelial, like this one is epithelial, but has very low Wnt signaling. So that Wnt pathway, which was characteristic of the Vogelstein model, is not there. And this is more of a metabolic abnormality. It's only 11%, but this is predominantly KRAS mutant. You will know that, well, you may know that KRAS mutated cancer is a huge unmet need. And there is a specific task force set up by the US NCI to try and find a way into therapeutics of KRAS. And it won't be, I think, by targeting KRAS directly, but it'll be finding the ways that different biologies are associated with it and targeting those biologies. So that gives us new insights. And this was another group that we knew nothing about. We knew nothing about this group of 20% of patients. These are actually the bad actors. The model we've all been looking at, the Vogelstein model, actually they do very well, those patients. They're not the problem. It's this lot that are the problem, and we're only just beginning to see them. So as we think about stratified medicine or precision medicine, what biological model are we using to stratify people? Is it a gene sequence mutation-based model, or is it a more biologically driven model? And that's going to influence the way we actually stratify patients and design our trials. On top of that, there's about 20% who don't fit. But when we come back to the clinic, the trial but 3 ago that we ran was called the COIN trial, a very large trial with 2,500 patients in it. And of those, we characterized about 2,000 patients with a simple set of genetic mutation markers. And you can see the landscape at this level. And you can see there's this big lot, 40% KRAS mutation. There's another lot, small group of NRAS mutation. And these 2, if that's come up, I don't know whether it does show, those 2 now come together to be the RAS mutant group, which are not sensitive to EGFR targeted therapy. The BRAF mutant group is a very important group with a very poor prognosis, and I'll talk more about them in a minute. The PIK3CA mutation group, another group here which overlaps, half of them have RAS mutation and half of them don't. And then scattered through that macrosatellite instability group, the group that was on the left-hand column. And outside of that, a group that are wild type for these different molecular markers. So that's the sort of clinical stratification basis that we're working on at the moment, rather than the comprehensive genetics or gene expression. And the ones in pink, as I said, are the ones that are not sensitive to RAS EGFR mutation. Now, as we've done this, and I'm going to be talking about various ways in which we've learnt that have led us to the ability to deliver a stratified medicine trial. And one of these is about developing biomarker capability. And we've worked with 2 labs in Leeds and in Cardiff, where there's been a lot of work on previous colorectal trials. And then in our previous trial, Focus 3, we got them working together. And I think the concept of 2 or more labs working together, sharing quality control methods, is a very much more robust way of doing it than just relying on one lab. So Focus 3 was where we were 3 or 4 years ago, and this was the feasibility study for Focus 4. And when I first presented this at the National Colorectal Meeting, I presented it in a different way. It subdivides people into 4 molecular groups. And in each of these groups, you can be randomized 3 ways. And that makes a 12-arm trial. And there was a gasp in the audience as the research nurses thought, how on earth are we going to explain that to people? This is a first-line metastatic disease trial. So can you just put yourself in the clinic room a minute? Here I am talking to you, and I've just explained to you that you've got incurable metastatic cancer, and you are going to die. And then I tell you in the next breath, would you be interested in a 12-arm randomized trial? You know, your head is not there. So how do we begin to explain this? And I want to give credit to Malcolm and Jan Pope. Malcolm was a patient of mine a number of years ago and his wife, and they have been working with us over a number of years. And we just simplified the concept down to the absolute bare bones using this diagram. We want to do 2 tests on your cancer, and they're going to be positive or negative. And that means your cancer could be one of 4 groups. Will you give us permission to do the tests? That was it. People could get their head around that. So on that terrible day, when we had that first oncology consultation, we gave them this patient information sheet, which enabled them to register and allowed us to get the process going in the lab to do the diagnostics. And that was the diagram that we used. Next week, when they came back to the next clinic, when we were telling them about chemo and all that entailed, we gave them the whole lot about what's a randomized trial and what the toxicity was likely to be and a bit more about it. 2 weeks later, when we had the results of the marker tests, we gave them just the bit that was relevant. So if they were in molecular type 1, then this was the trial that they were going to be going into. So they didn't need to know about what everybody else might be getting. They just needed to know about the bit that was relevant for them. So they got the relevant patient information sheet. And then when they were randomized, they got the details about the specific treatment, which one of these 5 treatments they were randomized to. And we asked them how they got on with this, whether they understood it. And so these are the people who were in that situation. And this is what they said 2 weeks later, that most of them, half of them, more than half of them, understood fully what this trial was all about, and the others understood mostly. And you can see very little level of not understanding, because, as our previous speaker said, it's actually a very simple concept. It's about trying to fit the treatment to what their tumour tells us. And people like it. So what we learned from Focus 3, one of my colleagues, John Bridgewater, a very experienced oncologist, said this is the first trial ever where every patient has said yes. Because it's simple and it fits to them. So though it's complicated, they loved it. We recruited 240 patients in a year, despite the complexity of the study. Our primary endpoint was to get the results back to the investigator within 10 working days. So if you're doing a genetic test, can you turn it around fast enough? Because these were patients who were waiting for their first-line chemotherapy before they started. Now, in fact, 2 weeks is about how long it takes us to set it up in the clinic, so that's okay. So a 2-week delay was okay. In fact, only 74% of the results were available within the 10 working days, but 99% were available within 21 working days. And actually, when we looked at specific patient delays, the clinical delays and the waiting for the patient to be fit for chemotherapy was actually completely dominated the picture, and the waiting for the biomarker results really made no impact. So we took this as a proof of principle that we can undertake complex prospective biomarker-driven trials in the NHS in the UK. And that was what led us to prove the feasibility for the Focus 4 trial. So what have we learned about biomarkers? The colorectal cancer is complicated, but there are some subgroups that we can define and we can measure. We can do that using 2 labs. We can do it in a month. 2 labs working together is a pretty robust way of doing it. The second point to think about in these trials is, when do you intervene? So you have to think about what's the patient pathway, and we want to be as early as possible. So a standard paradigm for doing cancer trials is to wait until they failed everything and do it at the end. Now, we don't want to do that, because that way resistance has emerged, we've got multiple resistant clones, and we fail a lot of drugs that otherwise might be successful. So going back a decade, 2002, we published the results of a previous trial which showed that giving patients a time off chemo was not detrimental to them. And we wanted to test that in the modern era, that here's the standard of care, continuous chemotherapy until progression. Could we give them a holiday without affecting survival? And we did another trial, which I'll come back to, the COIN-B, which used that interval. So using the interval. This was the result of a very large trial called COIN trial, published in The Lancet in 2011. And you can see that the people who had the intermittent treatment in red, their survival was minimally different. There's about a 6-week difference. But it did fail our non-inferiority boundaries. So there's a very small difference. Quality of life was better. Patients loved their holidays. And we looked at reasons why this might be different. And here's the forest plot, and you can see that there's one predictor here which was statistically significant. Platelets at baseline. It's an odd thing. We get it free. It's a free biomarker. It's something we know all the time. And it possibly is a surrogate for something going on in the tumour microenvironment. But if you use that, people who have normal platelet counts have no deficit from the holiday, and that's about 3/4 of the population, whereas those with raised platelets have a 5-month deficit. So in Focus 4, we've excluded patients with a raised platelet count. In COIN-B, so this is then testing a proof, so we've established that we can find a group of patients, a large group of patients, where there's a good window for a stratified trial early in the course of the disease in colorectal cancer. Now we've done a test of, can you test a specific molecularly targeted therapy in a biomarker-selected cohort, uses the interval, and can you measure benefit? So this was COIN-B. So this was a cohort of patients with RAS wild type, where we were using cetuximab, an EGFR, a monoclonal antibody. And you can see that those patients who had the cetuximab through the interval as a maintenance therapy had a significant improvement in progression-free survival in the interval, and indeed survival. So we've learned something about finding this slot in the disease process where we can use this. I'd like to move this further forward. I actually now want to do this in the initial diagnosis at radical therapy. This is when we need to do it, so we can make an impact on cure around the use of radiotherapy, surgery, targeted drugs immediately at the time of first diagnosis. But this is where we are at the moment. So you can see those. We've proven the principle about using the interval. So on that basis, we designed Focus 4. And you can see that a lot of support here. There's a fantastic Focus 4 team. We have 2 of them in the audience. Louise Brown is our senior statistician from the MRC Trials Unit, and Jane Beattie. Where's Jane? Jane's over there from the Clinical Research Network, integrating pathologists, trialists, oncologists, patients, network people, data managers, etc. And Focus 4 is an integrated trial program which I'll design to exploiting that window of opportunity, testing novel agents before resistance has occurred using modern statistical designs, not Bayesian but frequentist. Okay, this is the design. So here are patients. Here we are. You're back in the clinic, that first consultation. And we're asking patients if they'd be happy to register for a trial. And then we're starting them, in fact, on standard chemotherapy for 4 months. All of them go through that, during which time we're doing the biomarker analysis. And that's our biomarker panel, a mixture of gene mutation, some mRNA, and immunohistochemistry. Nothing terribly complicated. And that, at the end of 16 weeks therapy, we allocate them into one of these 4 biomarker-defined cohorts. And then when each of those, they will be testing a novel agent in that interval, bearing in mind these are patients in whom it is safe to have time off therapy. So the placebo is no treatment, the control arm is no treatment, and the therapeutic intervention is the novel therapy. Now this is different from what Ian was describing beforehand. We're not just using this trial to try out patients, new drugs against patients. We're actually testing how much difference that drug makes against what would happen without that drug. And that's important, and I'll show you why on the next slide. The primary endpoint is progression-free survival in the interval, and then because you haven't treated them to progression here, you can restart first-line chemotherapy. You can use that principle of intermittent chemo to go back on. We're going to be rebiopsying at randomization and on progression. It's optional, not mandatory, but we'll be trying to get as much of that as we can. So I'm going to be giving you 7 principles which were published in a JCO paper about a year ago. And the first principle is to evaluate multiple treatments and biomarkers in the same protocol. So there's several things in that principle. First of all, this is the patient perspective. Patients aren't interested in just having access to the one targeted drug that company X is wanting to evaluate. They want a treatment that's best for them. So you need to have a range of options available fitted with the biomarker profile of the disease. So multiple treatments, and that means multiple biomarkers, and that means all of these biomarkers will be at different stages of evolution. So RAS is nailed down. RAF is emerging. PIK3CA, we don't know whether that's a relevant biomarker at all, but we're having a go with it to see whether it does work. So you'll have different stages of development of the biomarkers in the same protocol. Regulatory issues, we had to fight with the MHRA to allow them, to get them to agree with us that this was one protocol, not 6. But we have got one number. Every one of these new that we bring in is a protocol amendment, not a new protocol. Second principle I've made is, I've already talked about, investigate new treatments in the earliest and most likely responsive settings that are clinically feasible. That's very important for cancer. It may not be for other drugs, of other diseases. Okay, then we've got a bunch of new treatments that we're putting in here. And that was what we went to the funders with. At the moment, we've only got these 2 open, and I'll tell you a bit more about the way that's changed over time. But the third principle is used randomized evidence with a control group for each biomarker treatment cohort evaluation. And the reason that's important is this graph. So BRAF mutants have a median survival in this cohort of 8 months. RAS mutants, 14 months. All wild types, 20 months. So if you just looked at it without a control arm and you had a result of 14 months, well, what would that mean? If you just got the mixture, you have to randomize, I believe, to get robust data of efficacy. And then how do we analyze this? Well, this is a complex statistical plan with 4 stages. Stage 1, an interim analysis for safety and lack of specific activity. So we're looking to throw out hypotheses that are failing. If they do fail, we can bring in new hypotheses. But if they pass these first 2 stages, then we're pushing on to a stage 3, which is like a phase 3 trial. But we're also opening up this group over here, because we've taken a hypothesis here. We're testing, we believe that this biomarker defines the group who are sensitive to this treatment. What this enables us to do is to test the biomarker. Is it true to say that outside of this biomarker selective group, this treatment has no activity? Because in fact, many of the treatments may well have activity across other cohorts of patients. So we're testing the drug and we're testing the biomarker. And we're doing that sequentially when we've got an indication that the drug has at least useful activity, and we can push right on through to a full stage 4 analysis for overall survival if we get agreement. So the principle here is that in initial stages you're assessing each treatment in the presumptive biomarker-enriched subset, but without assuming in the design that this association will be confirmed at later stages. So you're testing the biomarker as well as the drug. So this is the sort of statistical plan which Louise generates for us. And you can see that within each of the cohorts, you've got an allocation ratio, you've got the different stages, you've got the target hazard ratio, which you can vary, you've got the timing, and the sequential numbers. So the principle here is you want rapid evaluation of each new treatment and flexibility, targeting a reasonably large effect size, discontinuation if you're not winning. So it's highly flexible, and in fact, we're flexing this to the max, as you will see. So let's just take an example of the BRAF cohort. So this is a really important cohort. It's only 8%. It's that worst prognostic group. And this is the group BRAF, I'm sure you're all aware of. This was the first gene that came out of the Cancer Genome Project on this site here, present in 50% of melanoma. And within 8 years of having described the gene, there is a targeted intervention in the clinic. And vemurafenib, targeting BRAF mutant melanoma, has had dramatic effects in the clinic. But when you take it into colorectal cancer, you don't see anything. Why not? So we thought, well, if you combine it with an inhibitor, the theory is it signals down this pathway. If you block it, you'll get benefit. Doesn't work. Block the next one in combination. It still didn't work in colorectal cancer. Then some lovely work published in Nature and elsewhere showed that actually you were getting signaling back through the EGFR pathway, and that was signaling down the other pathway here. So, and then when you combined an EGFR inhibitor into this doublet or triplet, you got benefit in the lab and to overcome the feedback. The triplet therapy of a BRAF, a MEK, and an EGFR inhibitor has been tested, was published in ASCO last year. That triplet is safe, remarkably, and that's going into focus for next summer. So there's been a delay while all of that biology has been elucidated, while the triplet monotherapy has been sussed out. And if this is the data from Gary Middleton, my colleague who presented at the ASCO discussion, and here's the triplet. You see only 15 patients have been treated at that time globally, but there's a 40% response rate using that triplet in this very non-responsive group and 80% disease control. So we're really excited about this very cutting-edge triplet treatment coming into focus for as one of its definitive methods of evaluation supported by GSK and Amgen, which certainly wouldn't have happened without this sort of trial. Just to go on, one needs to think about the way, because these cohorts are overlapping, you have to think about what's the sequence of allocation to the strata. So we're taking the BRAF off the top, then this next stratum, and going on that way. You have to work that out. And the principle here is to allow the possibility to refine biomarkers through the course of the trial. Now that's a tricky statement, because of course your biomarkers do need to be consistent and reproducible. But if you're going to introduce new biomarkers, that might be to introduce a new subset, which isn't so difficult. But if you're going to change the biomarker that defines a cohort, you've got to make sure you're doing that with a lot of quality control. So we're working between the two labs, looking at comparisons between the Leeds and the Cardiff lab, and particularly immunohistochemistry, which for instance we're using for P10 expression, very tricky to get working right in two labs, but needs to work together very closely. So where are we now? Well, so the triplet, the BRAF triplet will be coming in next year. It's been found in colorectal cancer that despite our lovely theory that PIK3CA mutations antagonists just don't work in colorectal cancer. It's too complex a biology. And there's a very interesting epidemiological study that aspirin does work in this cohort, and we're going to test that. But we want to be able to introduce new cohorts, so new agents coming along, in this case the ATR inhibitor, identified for patients who have lost ATM, the parallel pathway. This is a synthetic lethal approach. We've identified where this sits. It comes into this small group here, largely out of the all wild-type group. So the possibility of introducing a new biomarker and a pairing into the overall program. So those are the 7 principles that we've established through this trial. Now, of course, this trial also gives fantastic opportunities to learn. And so if you look at the baseline biopsy, the biopsy at randomization, the biopsy on progression, tremendous opportunities to identify, to refine the biomarkers, to help us understand mechanisms of resistance. And I'm not allowed to say this, so Jonathan, please close your eyes for the minute, but we have been shortlisted for funding on the on the MRC StratMed program, and we'll be particularly focusing on the BRAF and the all wild-type cohorts in this group, hopefully when that's confirmed very soon. We will also be collecting germline DNA, and we're one of the gel pilots that will be correlating the germline DNA and getting whole genome sequencing where we have sufficient sample. And then the whole question of monitoring patients and the sexy field of circulating tumour DNA. We're working with the Rosenfeld lab and their spin-out Innovata. And hopefully that will give us the opportunity to overcome some of the problems of sample selection and heterogeneity, because you're monitoring it sort of non-invasively through the plasma DNA. So that brings it to an end, really. There are the 7 principles, which I will leave up during discussion. But just a final point is just this is a huge team effort. I'm here talking to you today, but my co-chief investigator, Richard Wilfston from Belfast, who chairs the clinical studies group, each of the cohorts has its own chief investigator, the patient reps, the biomarker specialists, the MRC clinical trials unit who are running the trial, the research networks, which we were hearing about earlier, the experimental cancer medicine centres, where we will be referring all the patients who are having complex triplets, for instance, like in the BRAF, joint funding, CRUK and EMI, now to add MRC Escort, the pharmaceutical companies involved, multiple HEIs, and widespread translational science collaborations. So it's a huge UK effort that we're on the way to delivering. Current status is we have about 40 centres open, we have about 80 patients registered, and we're just working through the workup of the trial. Thank you very much.
Thank you very much. That's a very exciting trial design. Any questions? Eddie?
Yeah, I think you answered one question I had, which was when you're looking at almost a clinical stratification, a biological stratification, it was the role of something as simple as the circulating tumour DNA, because that gives you obviously limited biological information. I think you've answered that. The next 2 questions are around going through multiple patient information sheets. Is that something that's transferable to other therapy areas, or is this highly specific to the very informed cancer patient?
I think it depends on your trial design. So in Focus 4, we actually only have 2 patient information sheets at registration and randomization. So I think you just need to think very hard about what is the patient pathway going through this process. And when was that upfront trial and intervening in that highly emotive consultation, we wanted to strip it out and make it as simple as possible. And then the other information could come later. We're very aware of the emotional content of what we do and the information overload. So I think it depends on what situation you are dealing with, your trial, where you are recruiting, and what you have to accomplish in that time.
And the other one's a simple one. It's about the cost per patient of running such a trial.
Right. Well, the grant we have is 3.6 million from the 2 joint funders, and that's not enough. We're asking for pharmaceutical collaborators to be adding an extra 2.5K per patient, which isn't much from a pharmaceutical company side, much cheaper than it would be. But it's, and that's without any of the translational research funding. So it's sizable grants, much bigger than the standard clinical trials grant. Hi, Matthew.
Hi, Tim. Thanks. It was really interesting. I was just thinking about the practicalities of this trial. It's got a lot of arms. You're randomizing these patients. I was just wondering if you could kind of speculate for us, is this feasible in other cancer types where you may have potentially a requirement for more arms if there's more stratification, or where there's smaller patient populations? I mean, you know, it's a beautiful model, but colorectal cancer has very high incidence. And do you think you can roll this type of model out across other tissue types practically and deliver in a reasonable time frame?
Okay, so AML is a much rarer cancer. And basically, I've nicked this design from Alan Burnett and the AML team. So what are the elements of it? You're taking a biological sample, and you're initiating treatment while you do the biology. And then you're stratifying. And so you can go down to as rare a subgroup as you want. The question really comes, what is the limits at which point randomization becomes too complex? Or it becomes
statistical
too slow.
Power as well.
Yes, the statistical power, that's right. And so we're certainly we can do this down to subgroups of 8%. We've looked, Lou, have we looked at less than 8%? In MSI down to, yeah, we've looked right down to, well, certainly 5% is MSI. Colorectal cancer is relatively common. I think if you go down to rarer tumours and where you're talking about 1 or 2%, then I don't think you can randomise at that level. But you could still filter that into a design where in those cohorts you're just treating. The challenge to you is, well, how do you know what the untreated prognosis in these patients? Therefore, how will you tell whether it's really doing something? I mean, if you get responses, you can tell there's activity, but you don't always get responses. You may get disease stabilizations, and you really don't know how much that's being driven by the disease biology, the inherent disease biology. So I think we need to try and build in randomization where we can, recognizing that that's not always possible.